Thank you all for joining today. Have you ever wondered what invisible critters are below the soil surface and what they do? Today, our speaker is Dr. Willm Martens-Habbena, and he's going to give us some insights into underground ecology. Dr. Willm is an assistant professor at University of Florida. He's a microbial ecologist. He received his doctoral degree at the University of Oldenburg in Germany and was a postdoctoral research associate at the University of Washington. Current projects he's working on include carbon and nitrogen cycle linkages and characterizing soil, water, and microbe domains at the DeLuca Preserve. Recently, he received grant funding, yay, to expand his work to Archbold's Buck Island Ranch to study the impact of different rainfall scenarios on microbes and greenhouse gas emissions. So today he's going to speak about microbial nitrogen cycling in subtropical and tropical soils of South Florida and its links to soil carbon storage. So please welcome Dr. Martens-Habbena. Thank you.
Well, this could be really good. Thank you so much, Betsy, for the invitation and for the probably most comprehensive and formal introduction I've received for a seminar I've given. Yeah, it's great to be here. And it was great to see the ranch and all the different things that are going on the ranch. And I'm looking forward to spending a little more time tomorrow here. Yeah, I'm a microbial ecologist, and I appreciate that none of you probably works or thinks about microbes more than is this food safe or not. So, but this seminar is for you. So I've given this kind of seminar, I don't know how many times. So if you have questions, if you don't understand something or you want to know more, just ask. I know all this already. I want to start by introducing the people that I'm actually talking about or whose work I'm talking about, because I'm mostly sitting behind my computer just like this. Now I'm supposed to use this. So my graduate students, Claire, Shannon, and Jonathan, who all were most inclined to do lab work. They didn't want to go do much field work when it rains in Florida. Then I got lucky to hire a postdoc, Drin, who turned out after 2 field trips decided he also likes bioinformatics a lot. The only person who really likes to go out into the field with me is my wife, Julie. She was just part of the party here when Claire graduated last summer. And Uli Stingel is our colleague that I'm sharing the whole lab space and lab with. And really the work I'm going to talk about is Drin's work, some of Claire's work, and some of Jonathan's work, and some of it that Shannon is now following up on. But basically, the motivation for what we're studying locally is coming from what maybe some of you know is how water flow and water management has changed the Florida Peninsula and especially the southern part of it. And really what I found most intriguing is that so on the historic water flow on the left, that was like 120 years ago. 120 years ago, when a raindrop would fall in Orlando, the only way it could find its way to the ocean was to Florida Bay, which is way down south here. So the water mass that was basically forming a humongous wetland or connected wetlands here in the upper part, and then here one large contiguous wetland south of Lake Okeechobee, was about 3 times as much as is going on now. And this has changed because of humans coming in and trying to do agriculture and sell land and do business. And what they did was building canals, lots of canals that lower the water table and drain water east and west. And so now 60 to 70% of the water that is generated or like precipitation is now draining east and west. And that completely changes the downstream systems. And so we do some work here in an agricultural area south of Lake Okeechobee, which is a really, or is one of the most productive agricultural systems in the continental US, because it's basically 10,000 years of carbon accumulated in a wetland that now is steadily oxidizing and releasing all the nutrients that were trapped in it. And we do some work down here in the actual Everglades, in the Everglades National Park, as the as part of the long-term ecological research down there. But I will, in terms of field work, mostly talk about work that we do here in the agricultural area. And then the other motivation for specifically working on nitrogen cycle microorganisms is that we have not only engineered the Florida Peninsula, we have also engineered agriculture quite a bit. And by doing that, specifically or most pronounced by using nitrogen fertilizer, we have over the past half a century, primarily, doubled the amount of reactive nitrogen on Earth. So if you count all ammonia and ammonia nitrite and nitrate, these inorganic nitrogen molecules, if you count them up, there were half of them, half of it on the planet about 50 years or 60 years ago. And most of that has changed due to industrial nitrogen fixation, Haber-Bosch. And Haber-Bosch consumes some like 5% of global energy consumption. So on the one hand, we use a lot of energy to actually fix that nitrogen, and then we use it in agriculture. But it turns out that only about half of that nitrogen that's actually applied as nitrogen fertilizer in agriculture is actually ending up in any kind of crop. The other half is going elsewhere. And some people who are better at understanding this problem or this complex globally think that actually the modifications that we've made by introducing so much more nitrogen into the biosphere is considered one of the largest threats, larger even than climate change and biodiversity loss. And here the two or three like components that are highlighted here in red are highlighted in red because people think that we've gone so far, we've modified the nitrogen cycle so far that within our lifetime, or at least the next century, it will not bounce back to where it has been, or it may never bounce back to where it has been. So I talked a little bit about this this big picture problem, and I want to introduce a little bit of the microbiology that is driving all the processes associated with nitrogen cycling. And it turns out all reactions that occur in the nitrogen cycle are driven by microorganisms. And naturally, the first step would be nitrogen fixation, and that is driven by microbes either freely in the soil or in the sediment or in the water, or in root nodules associated with plant roots. That would then turn into organic nitrogen, and when the plant biomass dies off, it would turn into ammonia. And that ammonia can then be either re-assimilated by new plant material, or it can be oxidized to nitrate by microorganisms. And the nitrate can be taken up back into plant material, or it can leach, because nitrate is more water soluble than ammonia. And so when it rains, I'll show you a few examples later, that water soluble nitrate washes out much more easily than the particle bound positively charged ammonia. And then nitrate can also be, by microbes again, denitrified back into N2. And so if you just look at this figure and say, okay, if we have increased this pool twice over the past 50 years, so all that needs to happen is really just this needs to go twice as fast, and this needs to go twice as fast, and we'll be good. And if we figure that out, then there will be some Nobel Prize given for it or something. But the problem is in the details, and that's, what are these microbes doing? How are they working? And in agriculture, actually, we don't want these organisms because they turn things into nitrate and they leach. And so if you've applied fertilizer in December or January, and these bugs are active, then in May, everything is nitrate, and then it starts raining. And then the nitrate is feeding the algae on the Atlantic side or in Tampa Bay, but it's not feeding the citrus trees or sugarcane or whatever you're trying to grow. So we're trying to prevent this in agriculture. And so then this here is actually not really happening, or if it's happening, we don't actually exactly know when it's happening. And so that is kind of what we're trying to figure out in this new project that we're engaged in. Okay. Okay, I just explained this, and really the problem is, where is this nitrogen going, and can we influence where it's going? Do plants have an influence on where it's going? Do plants have an influence on the ammonia oxidizers and so on? And I just explained this already. This is basically globally what happens in Florida. The algae blooms on the coast. It's not a Florida problem, it's a global problem. It's much worse actually in parts of Europe, because the coastal areas, the water circulation in coastal areas are much more restricted. And so the water just stays in those bays, and they get really badly, you get really bad algae blooms. And of course, in some parts of Asia, Australia, these are hypoxic zones. And these areas become hypoxic because there's so much algae biomass produced by removal of nitrogen limitation that you get much more algae biomass. And that algae biomass in late summer runs out of nutrients eventually and dies off. And then all this biomass gets decayed by microbes and then causes oxygen depletion. And that is what people actually notice, because then they see dead fish, they see dead zones where there used to be fish, there is no fish anymore. Sometimes trapped fish, because they are trapped between anoxia in small areas. But all of these phenomena are, at the end of the day, are all connected, and they're driven by us having introduced more and more nitrogen, at least in part. There's other reasons, other nutrients and so on, but from a nitrogen cycling perspective, that's the main driver. And these processes also contribute to greenhouse gas emissions, because both nitrification and denitrification produce nitrous oxide, and significant amount of it. In fact, we think that some 70% of N2O emissions from agriculture are from nitrification. So the idea is, let's shut this down, and or maybe not. So I'll get to that a little later. But I want to introduce a little bit about the microorganisms that we think are involved in this process. And some of them we've known for more than a century. In fact, Sergio Vinogradsky in 1890 published about some of these organisms. And at the time, he already knew these bacteria these genera of Nitrosomonas and Nitrosococcus. Nitrosospira was found later. But basically, for a century, we knew the bad guys, basically. They oxidize ammonia to nitrite, and then there's nitrite oxidizing bacteria that oxidize nitrite plus to nitrate. And then just when I was finishing my PhD, I had learned all this basic microbiology. It turned out that, in fact, there is also archaea that can do the very same process. They also oxidize ammonia to nitrite, and then there's nitrite oxidizing bacteria that oxidize it further to nitrate. That's what I ended up spending most of my postdoc time studying. And it turns out that about 30% of the microbial plankton in the oceans do this. 30% of the microbes in the ocean are archaea, and almost all of them, there's a few smaller lineages that seem not to oxidize ammonia, but the vast majority of them actually are very, very good and very efficient at oxidizing ammonia. And in the ocean, that means fish are happy, corals are happy, sponges are happy. Actually, some corals and some sponges have these archaea as endosymbionts to detoxify the ammonia that the sponge produces and turn it into nitrate. So if you take a water sample in the ocean, almost anywhere, the primary form of inorganic nitrogen is always nitrate. In soil, it's a different story, but we'll get to that in a moment. And then I was working on these archaea, and then in 2015, a group in Europe found a third group of microbes that can oxidize ammonium. And these guys, actually, they were known to oxidize nitrite, and then were found to also have the genes and the biochemistry to oxidize ammonia to nitrite. So these are kind of do-it-all nitrifiers. So now, basically in the past 15 years, we've gone from 3 genera of bacteria that we knew are doing this to this diversity and some global estimates say like 5% of all microbes on the planet do just this. They oxidize ammonia to nitrite. And so it can't be that this is completely useless and that we should stop it entirely. So there must be some function. There must be, it must be part of the carbon and nitrogen cycle in terrestrial environments as well as in the ocean. Here are some electron microscopy images just to introduce some of the bugs. So this is an example. So this is an individual cell here of a terrestrial, a soil archaeal ammonia oxidizer, and this is the marine ammonia oxidizer. So these are the 2 archaea, and then all of these are the bacterial ammonia oxidizers. And I tried to get them to scale properly. So literally, it's like David versus Goliath. And so the first thing that comes to mind when you see this picture from a microbiology perspective is there's something different about surface to volume ratio. And logically, how that would translate, if all the enzymes were the same, they have the same affinity, they have the same rates. If you just plug the same amount of enzymes in that nitrosopolymerase membrane or into this nitrosococcus membrane, then the transport efficiency or the rate of reaction should be faster with the smaller guy, because the small cell has more surface to volume and can just have proportionally, if you then add it up per gram biomass, for example, is there's much more surface area and much more uptake enzymes. And initially, this is exactly what we found. So here, so this is stuff I did during my postdoc, using, like, trying to grow these bugs in laboratory cultures and measure with electrodes how they would oxidize ammonia. And then you can, the basic idea is, or the basic outcome is, that you can put a so-called Michaelis-Menten curve through this. Have you heard of a Michaelis-Menten kinetics? Yeah, so it's like if you ask a biochemist and you have nothing else to talk to this person about, ask them about a Michaelis-Menten kinetics and they will talk for hours. There's all kinds of modifications of it, but it's basically the simplest idea is you have an enzyme and you have a substrate, and how fast is this enzyme turning the substrate into a product? And then if the enzyme doesn't change, then the concentration of the substrate makes it more likely for the enzyme to find its substrate, and then the reaction rate is faster. And that looks like this, basically. It's a saturation curve. And it turns out you can do this measurement with entire microbial cells intact with thousands of enzymes, but because the first enzyme is a rate-limiting enzyme in this whole process, you still see this kind of pseudo-Michaelis-Menten kinetics. Biochemists hate listening to microbiologists talking about Michaelis-Menten kinetics because they think they own it on enzymes. And we are not doing good science because we are measuring this like dirty in entire cells. But we can't actually isolate the enzyme. I'll get to that later. But basically, the archaea that we found first, and that is here, have an extremely low half saturation constant. That means the concentration of ammonia that's needed to go at half speed. Yeah. Instead of 65 miles an hour, you go 32.5 miles an hour. And how much gasoline are you using at 35 miles an hour, basically? And that concentration tells us something about when the organisms are actually good at something. And so if this is a very low number, like here in these archaeal ammonia oxidizers, these are all marine ones, that tells us they are very good at very low concentrations. And that is why there is no ammonia in the ocean. There's only nitrate, which is the end product of it, in the ocean. But if we look at the few isolates and the few strains that we have from soils, it looks completely different. These are the ammonia oxidizing bacteria that we know, that we've studied for almost a century. And these are the archaea that we know from soils. And it looks like they have very similar kinetic properties, which is cool based on these isolates, but it totally contradicts the genetic literature. So when people have gone out and just collected DNA from the environment and measured how many of organism A and how many of organism B do you find, that literature suggests that A or B are really good in cornfields and sugarcane and wherever people apply a lot of nitrogen fertilizer. And the archaea are much better at much lower inputs. And where the organic, or where the nitrogen is primarily coming from inorganic sources, from organic matter mineralization. So Jonathan in the lab tried to enrich some of these archaea. So we have this humongous diversity, And we have about 20 in culture. And those 20 are from like 3 different lineages. And there is like 10 more families of these archaea where we don't have any representatives. So what Jonathan did is to look at these different or try to enrich from soil directly under nutrient limitation. And what he found is that under whichever scenario he was using, the archaea were winning out. So there's something else going on that actually makes them more competitive under nutrient limiting conditions. And we're still trying to find out how that works. Or works more mechanistically. In the in a second part, we're looking at actual agricultural soil and trying to figure out how these archaea and bacteria interact in actual agricultural soil. And we went to this area south of Lake Okeechobee because it's this very organic, rich soil where farmers can grow crops without nitrogen fertilizer application. So it's a very active soil where the activities are high enough so we can measure rates, but it's not stimulated by fertilizer addition. So the community composition is skewed towards the archaea instead of the bacteria. This is Dr. Aaron Wright, and he's standing at one of these soil depth posts. And because this is Everglades soil and basically 10,000 years of peat accretion that oxidized away basically in some 80, 90 years. So there used to be like between 12 and 16 foot of peat, and there's about 1, 2, sometimes 3 foot left. So we did some time series experiments. The setup doesn't matter so much. I'm going to skip forward a little bit. And here is the nutrient dynamics that we're seeing in these soils. And here's particularly the nitrate. And I was talking about the high activities. There's a lot of organic matter mineralized, and these different plots are all different treatments. Plot 1 is unmanaged. Plot 2, 3, and 4 are different kinds of rotations with fallow treatments. And the 5th plot is growing sugarcane. And what you can see is that in the spring, in the early spring, there's a lot of nitrate in these soils, and it's actually produced by nitrification. And then as soon as the rainy season starts, the nitrate disappears within almost a month, like between our samplings. So we think there's probably a lot of microbial denitrification going on just in this short period of time, but there's still a lot of nitrification going on. And let me skip forward here. And these are rates that we measured in the lab, where we basically take soil and we just watch it and measure over time how much nitrate accumulates. And in blue, you can see the net. That means we are not adding any nutrients. And this is basically the speed at which microbes turn ammonia into nitrate in the soil. And to our surprise, these rates were higher in the plant-covered plots, in the native unmanaged one, and in the plot that was growing sugarcane, whereas the fallow plots had lower rates. So this could be because of the time point that we were actually measuring, because it was like right after harvest. And these had been fallow for a while. But it could also be that there's something going on, some interaction between the plants and the microbes, where the plants are actually trying to get the microbes to be more active and feed root exudates into the community that then stimulate microbial activity. Here on the right, you can see the nitrous oxide, does this come through? Oh, there should have been a gray block here in the background. I don't know where it went. I don't see it here either. But basically, the amount of ammonia that is oxidized to nitride and the amount of N2O that's being produced during the same time, you can correlate and make a mass balance. And based on that, you can figure out who was the active one. We know that the AOB produce about a per mil of N2O per ammonia or more, and the AOA produce somewhere around 0.3 to 0.7. So it looks like in these soils, really the AOA are the predominant ammonia oxidizers. And then we did a conservative mass balance calculation and say, like, we got the harvest stats from the farm manager and calculated what the nitrogen amount is that the sugarcane that was growing in one of these plots was building up, and came up with around 1 ton of nitrogen per hectare, 1 metric ton of nitrogen per hectare that is fixed into biomass by the sugarcane plant as a whole, including above ground and below ground biomass. And based on our rate measurements, nitrification in the soil during the same period of time would produce 1.6 metric tons of nitrate. And during the whole time, the ammonia concentrations in the soil stayed pretty low. So we think that the plants may actually primarily rely on nitrate in these soils. And the reason why we've never looked at this or never thought plants might actually prefer nitrogen or prefer nitrate is that when all the plant nutrition literature was established, the idea was there's only these bacteria, these few bacteria that are very rare in soil and need a lot of nitrogen in order to be active. But now, knowing that a lot of these archaea are doing the same thing, we think maybe there is something else going on. Maybe there's not just nitrogen leaching going on, but maybe these nitrifiers are actually involved in plant nutrition. And that this is basically how the nitrogen cycle in a lot of these soils functions. I don't know, how much time do I still have? There's no...
About 25, 20 minutes.
20 minutes? Okay. I got a little scared because it said 47 minutes here. And I was like, oh my God. Okay. So, yes, it's coming. So, then Drin, my postdoc, was interested in figuring out what are all these different archaeal lineages, and can we start to tease apart who might be interacting with plants or who might be just a nuisance ammonia oxidizer. And he looked at one of the marker genes that we use to identify the organisms, and that's the ammonia monooxygenase gene. That's the gene that encodes for the first enzyme in the pathway. And what this is just like the overview. Basically, what it's telling us is there is 1, 2, 3, 4, 5, 6 families of these organisms with each, I don't know how many genera and species in it, that are coexisting and active in these soils. This is just a more zoom in. We don't have to look at this in too much detail. I do want you to read these though. But the basic idea here is that we isolate DNA from the soil and we isolate RNA from the soil and we sequence both. And then from the bulk pools, we sort out how much do we get, how many of these sequences that we get back are mapping to this particular enzyme. And the sequences that we get are long enough for a genetic fingerprint, basically. So we can tell, it is this species and it is this active. The number of RNA sequences that we get tell us how much of this enzyme is actually expressed in the soil. How much are they actually using it? And so it turns out there's a mix and match. So here, for example, is one that isn't as abundant in the soil, but it is very abundant on the RNA level. This means this is not the most frequent organism among these ammonia oxidizers in the soil, but at the time of sampling, this was the most active. Whereas here's an example of someone that's more abundant, but less active. So there's all kinds of patterns that are happening that we're now trying to figure out and tease apart. But the basic result from this is that basically all of these lineages, all these 12 families are very likely all actively oxidizing ammonia, and they're actually really happy. So here we used, again, the same kind of idea. You isolate RNA from the soil and map these genes, and we compare it to laboratory strains. So species that we've cultivated in the lab, and measure gene expression in those. And so here in these key enzymes, you can hardly see a difference, which is very unusual, because usually the assumption is that in nature, in the environment, microbes, plants, animals, they're always food or resource limited. And here they might be resource limited, but they are still very active. And they are in the soil growing basically as fast as we can grow them in the lab under best possible conditions. So then Jonathan looked at some of his reactors that he had set up under these nutrient limit conditions, and he found something really cool, and that is here in this electron micrograph. So these are two of the cells that are growing in a biofilm in these reactors. And when we don't know exactly yet how they come to build these biofilms, like what triggers when they start making biofilms, but we can see that they form these networks of cells that can get like hundreds of micrometers to millimeters in size and form cell-cell connections. And so the crazy idea here is that maybe these actually function like mycorrhizae and interact with mycorrhizae and with plants. There's evidence that mycorrhizae are really good at phosphorus acquisition, but on nitrogen, it's wonky. Sometimes mycorrhizae are helping, and sometimes they don't. And we think maybe it's actually the archaea that are helping on the nitrogen side. So that is kind of work that Shannon is now following up on. Okay, one, I think, last little vignette I want to talk about is a little more really lab microbiology, where we're trying to understand which process actually is responsible, like biochemically, for the amounts of nitrous oxide that these bugs produce. So I said before, like the estimates are like anywhere between 70 and 80% of the nitrous oxide produced in agriculture is coming from this process of ammonia oxidation. And in one of the previous slides, I was showing this graph with different levels of N2O yield in these archaea and in the bacteria. And we don't really understand why that level is different in this between the bacteria and archaea. So Claire has been trying to understand this a little better, and I have to bore you with a little biochemistry just for a moment. And that is basically the basic setup of how these enzymes are arranged in the cell. They are in the cytoplasmic membrane, so in the membrane that surrounds the cell. And this is the ammonia monooxygenase. So that's the first enzyme that we were using in the soil as a marker to figure out who is who. And that's the enzyme that oxidizes ammonia to hydroxylamine. That's the intermediate that the microbes are using. And then the hydroxylamine is oxidized here by the second enzyme, hydroxylamine dehydrogenase, to make the nitrite. And so some people say, well, maybe it's a hydroxylamine dehydrogenase that is actually sidestepping some nitric oxide, and that is then oxidized to nitrous oxide. So that's the prevailing hypothesis. There is a second way you can look at it, and some of these bacteria have basically a partial denitrification pathway, and which is really confusing because that organism that is basically making a living by oxidizing ammonia to nitrite can now take that nitrite and reduce it to N2O. And now things become really dangerous because this is basically a greenhouse gas production machine. In this mode, basically 30, 40, 50% of the nitrogen of the ammonia that is oxidized to nitrite is then used and reduced to N2O. And so far, we have not found a bacterial ammonia oxidizer that has the complete pathway that can take the N2O and add the last enzyme to make N2 out of it. It's probably because this reduction pathway, the oxidation pathway and the reduction pathway would end up being a catch-22, like the energy that's in it to oxidize it, like the oxidation happened at certain redox potentials, and the last step, the N2O reduction, is probably at a too different or too low redox potential for that to actually work. So they might have had this pathway way back when and have just lost it because it's inefficient. But Claire was trying to mimic this in the lab by basically asking, in a field experiment, you would ask, what happens in the dry season in December, where the soil water content is somewhere in the 30, 40, 50%. And what is happening in the rainy season when the soil water moisture level is at 80, 90, or 100%, and things become quite oxygen limited, or can become quite oxygen limited. So these set these up and figured out how to grow these bugs in these flasks, measuring oxygen concentrations, to have some of them at these very low levels and some of them at happy saturated oxygen levels. Okay. So what she found out is that she looks at the oxygen consumption and the nitrite production over time. So these are concentrated cultures. Things go a little faster, luckily. So this is the measurement that take like 2 hours. And here in blue, you see the oxygen consumption. And in green and in orange, you see nitrous oxide and nitric oxide. And what happens under oxygen replete conditions is pretty remarkably boring because literally nothing happens. You add ammonia to it. Here she adds 1 millimolar ammonium. Here is 10 millimolar ammonium. That 10 millimolar is actually high enough to kill all the archaea. So if there is ever a cow pile, the archaea will never like that. At least in its initial phase. But these guys actually really like it. And what happens in these oxygen replete cultures, those are cultures that were grown at ambient, basically winter dry season. There is no nitrous oxide being produced under any of these cultures. But if she grows the nitrous ammonias under oxygen limited conditions, and it actually starts expressing this partial denitrification pathway, or in fact doesn't start expressing it, it actually always has it expressed. there is some nitrous oxide produced, but then in an ammonia-dependent fashion, under high ammonia input, there is lots of nitrous oxide being produced. And what that tells us is that oxygen limitation and ammonia saturation together are needed to trigger this intense denitrification. And what that does is pretty remarkable. So here on the left are these Michaelis-Menten plots. Basically, here's the concentration of oxygen, and here's the activity, the rate of oxygen uptake by the cells. So this is combining all the different experiments with different levels of ammonium input, right, in the oxygen replete cultures. And we see a beautiful, even a biochemist would like it, clean-cut Michaelis-Menten kinetic. But in the cells that were grown under oxygen-limited conditions, we see that at low ammonia concentrations, 1 millimolar, 2 millimolar, even a biochemist would agree that this is a Michaelis-Menten kinetic. But as the concentrations of ammonia go up, this is basically linearly decreasing oxygen uptake. So the lower the oxygen concentration becomes, the enzymes have a harder time actually metabolizing the oxygen. And why is that? Because one of the intermediates in the denitrification pathways, I think I had it here in orange is nitric oxide. And the nitric oxide is a very reactive intermediate that can bind to the enzyme in the respiration. So I'll skip one forward, maybe I'll go back in a moment. So what happens is that the cells are oxidizing ammonia, and as long as more ammonia becomes available, the pathway gets saturated, and the electrons that are generated from the oxidation process need to go somewhere. And because that's not happening fast enough, the enzyme that would usually reduce oxygen, the respiration enzyme that we have in our mitochondria and that these guys have in their membrane, that would usually reduce oxygen to water, gets blocked by nitric oxide. And it slows down. It doesn't kill the enzyme, but it slows it down significantly. And now they have a problem. They don't know where to go with these electrons. And now they are transferring them to nitrite reductase and nitric oxide reductase, producing nitrous oxide. And we can induce this in basically all the conditions, right? So the gene expression, actually, I try to highlight it here. The gene expression patterns between these two conditions, oxygen replete growth and oxygen limited growth, do not change. So if we took the RNA from the soil, we would not see much of a difference. So it is really the nitric oxide that, without any genetic regulation, blocks the oxygen reducing enzyme, and that's what pushes the excess of electrons into the denitrification pathway. And so that's why these massive N2O productions only happens in the first couple of weeks, 6 weeks, 10 weeks after nitrogen fertilizer application, and preferentially when the soil is like somewhat depleted, at least in oxygen. So actually the microbes get depleted in oxygen and get in this boundary condition. So to prove this, like in situ and figure out if we can look at this actually in the soil and figure out where are these microbes and are they associated with plant roots, and does plant nutrition or plant growth have any influence on it, or are they in soil particles, or where is this happening? This is work by Jonathan, my third graduate student, and he is trying to localize the enzyme in the cell. So it's like almost a biochemist project. But what he's trying to do is actually inhibit the enzyme with inhibitors we know that bind to the enzyme, and then use click chemistry to link fluorescent dyes to it, and then go to the microscope and look at it. So basically, he is having an active enzyme. He's inhibiting this enzyme, which with chemicals we know inhibit it, and then trying to add fluorescent labels to it, and then look in the microscope. And after a long period of testing, trying, and figuring out, he finally managed to make it happen. And so here is an example. I don't know how good you can see it on the screen, but basically he's using different concentrations of this inhibitor. And these are in red, or technically it's an overlay of phase contrast and fluorescence microscopy. But basically the red dots in these figures are cells that are labeled, that are fluorescing red, because we have labeled this enzyme in the cell. And here is a negative control, where we basically used unlabeled inhibitor first, and then ran the procedure, and that tells us that this is actually specific. So these cells here, they fluoresce because this ammonia monooxygenase enzyme is fluorescently labeled. And now we're trying to use this technique to basically, under ideal scenarios, take a soil sample and run it through this procedure, and then count in the microscope or look in the microscope, look at roots, look at soil clumps and so on, and figure out what the patterns are and where the organisms are actually located and active. And here is a control. So this is Pseudomonas strain, a very common soil bacteria that actually is denitrifying and is also capable of oxidizing hydrocarbons. So we wanted to use this as a negative control, and it turns out it does not fluoresce, so that's good. And now this is kind of like the downstream where we're trying to take this to basically sort out these nitrifiers and then identify them later on. Okay, and I think that's as much as I have. What can we conclude from this? I hope I think we know now that ammonia oxidizing bacteria can actually be part of a healthy nitrogen cycle in agricultural soils, and most likely also in natural soils. There is clear niche separation between the archaea based on nitrogen and phosphorus acquisition. I didn't emphasize or didn't go through this one graph with this cell cartoon and all these different enzymes. Turns out that between all these different archaeal lineages, the main difference that we see is that they are either good at low or high nitrogen concentrations, or low or high phosphorus concentrations. And with this matrix of enzyme activities, you can explain why these 14, 16 main lineages exist. So the kinetic characterizations yield conflicting results. The strains that we get from our nutrient-limited enrichments have much higher affinities to ammonia and are much better at low concentrations, no matter from where we get them. And AOB are likely the key drivers for nitrification-based N2O emissions. And I hope we now understand why. Okay, and with that, I'll thank my crew again, and I'll thank you for hopefully not running into too much oxygen depletion. Yeah, sure.
I just had a quick question. So you showed that there were some of those bacteria were present in seawater and some in the soil. Are there any evidence there are different groups in brackish water?
That's a great question. I think there are, and I don't know if I can go back easily. Give me 1 second, because so basically, there's like 2 or 3 families that we know primarily from the oceans, and now actually in our survey we find them also in soil, but at a much lower proportion. So they are mostly marine. And then in the estuaries, you have like individual lineages, but there's no clear family separation. It's not like, so the families that are dominant in soil, you absolutely do not find in the ocean or in estuaries, or at least not active. But the marine ones you find in estuaries, and you find them in even in soil. But there must be some adaptation that I'm not an expert on, so I wouldn't know.
Great job. So you mentioned that N2O is released when there's like oxygen depletion in the soil. So, like say, we repeat that in the soil gets flooded, like say in the wet season in these areas. Do you expect higher N2O emissions than that?
So, yes. So it's a tricky business for these microbes, because the ones that produce the nitrate depend on oxygen, right? Some of them, not all of them, have this nitrifier denitrification, this N2O like rocket system basically. But those are going to be active until oxygen is completely gone, whereas the other ones may pass out a little earlier, right? And then things shift to denitrification. That's when basically everybody else around in the soil is like, okay, now we are also running out of oxygen. And then wherever can, we'll start denitrifying. But it's when exactly that is happening and how this is coordinated basically in soil is not very well understood. And I think it's really this onset of wet season when the soil moisture gets high enough. And of course, the moisture gets high in the bottom first, right? So you can look at a soil that's on the surface still quite dry, but as the groundwater level rises, that nitrogen, that ammonia down there, that nitrate down there is starting to be denitrified. And then the thing moves up. So when you put a cylinder on the ground to measure anything, then what are you looking at, right? Is that N2O coming from like 2 meters down, or is that N2O coming from like right from underneath the surface? And when is it coming, right? So yeah, but in my naive, simplistic view, I think you could put a moisture sensor out and predict that as soon as moisture crosses the 80% water-filled pore space mark, you will get 5 days of denitrification, and all the N2O that's being produced throughout the year, or like 90% of it, is in this 5 days. And after that is fermentation, I think. That's also a great question. I mean, the simple model is, well, they just compete for the same substrate. But actually, it turns out preferentially, they found a way. I didn't talk about that at all, but they found a way to actually evade each other. So both of them can use ammonia, or at least representatives of both can use ammonia or urea. But it turns out the gene regulation in both is the opposite. So if you give them both ammonia and urea, the bacteria will take the urea and oxidize it to ammonia, and then use that ammonia and oxidize it to nitrite. Whereas the archaea will first use the ammonia, and only after they've run out of ammonia, then they'll switch and start using urea. And so I think, so the archaea, we think, are evolutionarily anywhere between 1 and 2 billion years old. And they have been in soils way before the first plant dared to put a root down. But the bacterial ammonia oxidizers are much, much younger. They are maybe 300-400 million years old. And so we think that, yeah, it's the bacteria that were basically having some opportunities. And then when the opportunity came to access urea, took advantage of that and found a better set of enzymes to actually do that.
Yeah,
sorry, I should have, I'm not sure if on Zoom. Okay, nobody had complained. I forgot to repeat the questions. Sorry.
So you mentioned that we went from like knowing the 3 bacteria do this to a bunch of bacteria and archaea that do it. And I was curious when they're prospecting for new species that do this, is it based on analyzing the sequence of the DNA, or are there more experimental ways to determine if it's a species is similar adaptation?
Great question. For the longest amount of time, we've done it by just looking for this gene of this first enzyme. And most of the time by looking for it with PCR. And if you do a PCR, what you need to do is you need to put 2 primers into the PCR reaction that are basically 2 pieces of DNA sequence that you need to know for the PCR to then give you back the sequence that was in between. So you're always basically like, you need some evidence to start searching, and that limits what you can find, right? And so now by using this shotgun sequencing, we're basically just sequencing bulk DNA, right? And what turns out, 2% of that bulk DNA goes with the archaea, and then some 0.1, sometimes less, goes with the bacteria. And now we can start searching just for this enzyme and find enzymes in this genetic data that maybe didn't match our PCR primers from before. And this is exactly how the third group was found in 2015.
How does shotgun sequencing work if not using primers?
So when you have, so there is like generations of DNA sequencing, right? So for the human genome, for the first human genome, they used a classical Sanger sequencing. What you need to have there is basically a PCR product of DNA that's the exact same sequence. And then you can basically do another PCR where the nucleotides are labeled by color, and then you run past basically a photocell, and each time a nucleotide is going by, you see a color. And so that's how they could do it back then. Now these newer sequencing techniques work by basically attaching the piece of DNA to a glass slide that you can look at in the microscope. And then they wash a reaction mix through. There's lots of variations to this, but the basic idea is like you wash a reaction mix through that only has one colored nucleotide. So let's say you wash a T through this, and whenever a DNA polymerase or ligase or incorporates a nucleotide, you got a light flash. And that light flash you can literally look at in the microscope in this particular spot. And because you are now just limited by the pixels of the camera, you can have camera systems that can look at billions of sequencing events basically at the same time. And that's why it's so much faster. And then you watch, you take a picture, then the next nucleotide, you take a picture, and a picture, and a picture, and a picture. And then the computer algorithm just goes back and says like, in this coordinate in this picture was a nucleotide. Where was the next nucleotide? And then it just goes through the stack of images at this particular spot. It's basically the same as if you would look with a satellite at a tree. And then you ask, when does a tree have leaves or when does it not? Like the idea is the same.
I was intrigued by your graph early on where you had the native plants and then the fallow fields and the sugar cane.
Okay.
And so you concluded that plants could interact with microbes and maybe stimulate nitrification.
Mm-hmm.
And I'm wondering if you think plants could differentially activate nitrification at like different levels. And if so, could we identify those species that increase leaching versus others? It's kind of, we found a similar outcome for phosphorus at the ranch, like comparing different species, and it looks like some of the plants stimulate phosphorus like availability, and then they are either able to take it up or it leaches away, and they do that at different.
Yeah,
like species are different in how they can do that.
Yeah. We don't know, but part of this grant is to look at basically the 200 most common plants in Florida and pick them out of the ground and look what the composition of microbes is in the root, along the root, and in the bulk soil. And can we tell that specific species have enriched for specific archaea? People have done this before, like we didn't invent this, but so often this is done by PCR again, and it turns out that the surveys that have been done didn't always use good primers that cover archaea as good as bacteria. And then you kind of get a very good picture for the bacteria. But if you are missing and not covering the archaeal ones very well, and they shouldn't be dominant. It shouldn't be like that the plant root now is covered with archaea. No, 90%, 95% is going to be bacteria. So if you isolate your DNA, your PCR will get you, in all likelihood, 99% of the bacteria and only maybe 1% of archaea. And so we're trying to do this slightly different to be more sensitive for the archaea.
I hope you use Bahia grass as one of your species. That's one of the ones that like seems to stimulate the release of phosphorus, I think, so it can take it up, but then by doing that, it somehow increases leaching of phosphorus.
Yeah.
It'd be cool to know if it does it to nitrogen too.
Yeah. Yeah, for sure. It's a lot of grass species have the ability to, or like some of the root exited compounds actually inhibit this very enzyme, this ammonia monooxygenase. So we're trying to, it is very possible that there is conditions when the plants actually do not want, if there is a lot of ammonia, the plant has no interest in looking for nitrate because it has to reduce it back to ammonia inside the root cell, right? So we think there is a window where this interaction might be important. It's not like I don't know. We speculated about this in a paper in 2021, and it took a couple of weeks. I got an email from a company in South America that asked me where they can buy these archaea because they want to now dump it into the soil, like into their agricultural soil to make it. And I was like, well, maybe we should have phrased this a little different.
Thank you so much.
Thank you.